DNA, the hereditary material. So DNA is the nucleic acid molecule that governs the process of heredity in plants and animals. Every cell in your body, except for gametes, contain the same DNA within its nucleus. That way the DNA is expressed, however, differs from cell to cell. Okay, so discovering the structure of the DNA. So it's important for us to go click on these two links that we find down at the bottom. James Watson, Crick, and Rosalina Franklin were three of the major players in the discovery of the DNA. So have a look at those links, some really good information there. So the structure of DNA itself. So going back to grade 9 science, we know that this picture over here is something that we've seen. We can see here that adenine and thymine are considered base pairs, and guanine and cytosine are also base pairs. Now, moving forward to Bio 30, we're going to look at more of a complex diagram. So DNA molecules form a double helix with a sugar phosphate backbone held together by complementary nitrous base pairs. The rules for DNA molecule include, first one, hydrogen bonds form between the base pairs. So remember, our base pairs are the AT or C and G because they're the pairs that go together. Adenine will always pair with thymine. Cytosine will always pair with guanine. Each base pair is bonded to a sugar group. Sugar groups are bonded to phosphates. The grouping that exists between a phosphate molecule, a sugar molecule, and a single nitrogenous base is collectively referred to as a nucleotide. And this is a picture that we'll see at the Bio 30 level. We can see the P's representing our phosphate, and we can see that we have our base pairs in there as well. Okay, so DNA strands are also said to be anti-parallel, meaning they run the opposite direction of each other. So the first point here, 5' and 3' ends run in opposite directions. Okay, so if we look over here, I have a 5 on the left and I have a 3' here. Now we don't need to understand why, and really we don't need to understand what that means, but what we need to look at is just understanding that we've heard of this terminology and know that it's anti-parallel, because this relates to some of our organic chemistry. Okay, this property has important implications for DNA replication and protein synthesis. So I kept this diagram in here for those of us that are chemistry people. You can have a look at this. But when we look at the 5' versus 3', like I said, it's based on our organic chemistry and where those bonds are happening. So if you have a look here and you're a chemistry person, I'm not going to go over the chemistry, but you'll be able to see from this diagram where things are attached, why we describe it as 5' or 3'. So first thing that we are going to talk about is DNA replication. DNA replication happens during S phase of interphase. A cell replicates in its entire genome. So what we need to look at is we have a DNA strand here. It's going to replicate into two DNA strands, and it's identical. So each strand of DNA serves as a template for the creation of its complementary strand. DNA replication is thus said to be semi-conservative. That is, each new molecule of DNA contains one strand of the original molecule. So looking at this picture here, is the blue is considered our old strand. So if we look, we had a single DNA strand here. And what happened in DNA replication is that our DNA strand is going to be separated, that those base pairs are going to separate. And if we look here, what happens is we have a new red strand that is going to attach to each part of the old strand. So what that allows us to do is that we have our old strand, it's going to split, and we're going to have that new red DNA attached. Okay, I also have you guys posted a DNA replication video Very important for us to watch because it's going to go through the animation, which is going to clarify this in great detail for all of you. Okay, so in DNA replication, step 1 is initiation. A group of enzymes known as helicases, that's important, bind to a specific sequence of a DNA referred to as the replication origin. The helicase is going to cut. It's going to cut that original DNA strand. The Y-shaped area that forms where the DNA is split is known as the replication fork, while the unwound region is known as the replication bubble. The single unwound strands of DNA serve as templates for replication. So this is what we need to look at, is that we have our original DNA, the helicase enzyme comes in and it cuts. So this here and this here are original strands. Okay, step 2, elongation. A primer sequence synthesized by the primase enzyme signals DNA polymerase III where to begin. So I know that's a lot of information, but I think our focus here needs to be that primase enzyme is going to be the starting point. DNA polymerase III begins to add nucleotides within the replication bubble, creating 2 new strands of DNA that are complementary to each other. So have over here, look at the picture, okay? And it's important that we do look at that animation video from the Amoeba Sisters because it goes through this in quite a bit of detail and it will help. Once replication is complete, the primers are removed by DNA polymerase. Elongation takes place in the 5' to 3' direction, meaning that one strand will always be leading and the other one will be lagging, okay? The Okazaki fragments of lagging strand are spliced together by an enzyme called DNA ligase. Think of this as the glue. So if we go back over here to our picture, our Okazaki fragments, this ligase enzyme is going to glue them together. As these pieces come in and find their matching on that original strand to create that new part of this strand, is that DNA ligase is going to act as a glue and bond that together. Okay, step 3, the termination. As soon as the newly formed strands are complete, they rewind automatically into their chemically stable helix structure. Replication proceeds until 2 new DNA molecules separate. The dismantling of the replication complex is known as termination. Now, a lot of information. This page is going to recap, and please, like I said, go back and watch that video with the Amoeba Sisters because it's super helpful. But this is going to help, and you got to write this in your notes, explain the DNA replication when we look at just the enzymes. Okay, your helicase cleaves and unwinds the DNA. The primase is acting as that primer to begin elongation. The DNA polymerase, okay, it's going to remove that primer. Okay, and the DNA ligase is going to splice together those Okazaki fragments in that lagging strand. Okay, so in this next part, we're going to move away from DNA and we're going to kind of look at how genes are going to express themselves. So what we're going to look at is we're going to start with DNA, but we're going to look at how that is going to transform into other forms that are able to be read and used throughout gene expression. So in order for a segment of DNA to be expressed by a cell, it must be first transcribed, and this word is really, really important, into mRNA, and then translated into a sequence of amino acids, which are a protein. This is referred to as the central dogma of gene expression. So have a look at this picture. This picture really is the best summary, is that we start with DNA, transcription occurs inside the nucleus, then translation occurs and we get what we call proteins, which I will refer to also as a chain of amino acids. If we go back to Bio 20 and we talk about those building blocks of amino acids, etc., this will make a little bit more sense to us. Okay, so what gene expression does, it allows different cells to express different genes. So although all cells contain the same DNA, they all don't behave the same. So one thing we need to look at is we've looked at DNA. DNA is what we've looked at in DNA replication. This is what we know that is inside a nucleus. But what we are going to look at is the transformation of that DNA into RNA, which allows it to be read in a different form, to express in a different way. So looking at a comparison, DNA has deoxyribose sugar, where RNA has ribose sugar. DNA has 2 strands, okay, which is a double helix. RNA has 1 strand. We also refer this to as single-stranded in some terminology. This is really, really important, is that it has in DNA a CGAT, but if we look at RNA, it has a CGA and U instead. So the big difference is a DNA has a thymine and RNA has a uracil instead. So that T is what's really going to be transformed into that U. Protein synthesis. Now I can go through this in detail, but this animation once again is very, very imperative for us to watch from the Amoeba Sisters. You may even need to watch it 2 or 3 times to understand this process. So please, you guys, it is so important that you pay attention to that one. Now, when we look at transcription, so we're looking at where this is not DNA replication. This is separate from DNA replication, but our transcription is going to take the information in a segment of double-stranded DNA is going to be copied into a single-stranded strand of mRNA. So this is the first step in protein synthesis. Very important, transcription, DNA to mRNA. Very, very important. So this picture here gives you a good visual of what happens, is that the top is our DNA, the bottom is a single-stranded mRNA, which we refer to mRNA as messenger RNA. So transcription takes place in the cell within the nucleus. Only 1 strand of DNA is transcribed into mRNA. This point is important, that we take the original DNA strand. So if we look at this picture and, okay, I guess you always go back to the original. I'll touch on that on the example. Transcription is initiated with RNA polymerase binds to the promoter region of the coding strand and begins to add the complementary base pairs in the 5 to 3' direction. Now, thymine is replaced by uracil, U's instead of T's. Once RNA polymerase reaches a termination sequence, mRNA strand is complete and can leave the nucleus to be translated into a protein. Okay, this part is important. Thymine is replaced by uracil. Okay, before translation takes place, the mRNA sequence is modified to remove sequence of mRNA called introns. Now, I'm not going to go over this in great detail. This is actually considered extra. So if you'd like to go over the introns versus extrons, you can read this, but it's not expected that we need to know this. Okay, I wanted you guys to go to the first practice 1 question. Okay, so the reason why we're going to jump here is that we're going to just look at transcription in, I guess, a bit of isolations before we kind of go on. So what we look at is the first thing it wants you to do is transcribe DNA into mRNA. So we know that DNA is found in our nucleus. So this is considered here a DNA strand. This is our double helix. Okay, so we look at this DNA strand and we can see that we have base pairs. So when we say our base pairs, we can see that CG, TA, etc., these are what we consider our base pairs. Now, when I look at this, what I like to do is I'm going to break them up into groups of 3, okay? And what I do is I draw a line to break them up into groups of 3 so that it's more manageable. Okay, when we look at coding these. Now, when I look here, we want to take them into DNA, the DNA into mRNA. And we know mRNA is a single-stranded. So looking at this, we are always going to go to the original DNA strand, which is this top one. Okay, so we are going to take that original DNA strand and we are going to turn it into mRNA. Okay, so we take this top row only because when we are going to transcribe the DNA into mRNA, we want to take the original and we assume that it's the top or the first strand. So the reason why I broke it up into segments of 3 is it helps us keep track a little bit. So when we look at mRNA, mRNA is the opposite of DNA, okay, except there are no T's and we use U's instead. So this first blue letter is a C. That is going to turn into a G. G. T would go to A. So the first 3 letters of this mRNA strand would be transcribed into GGA. Now let's go to the next group of 3. T would go to A. Now normally, okay, A pairs with a T, but in mRNA, there is no T. So I'm taking this first one, is that A is now going to turn into a U, and that C is going to turn to a G. Here, it's going to be A. Okay, this A right here, there is no T, so I go U. T would go to A, U, U. This T would go to an A. Sorry, G. That G would go to a C. That C would go to a G. Sorry guys, that's a G. T would go to an A, G, U. T would go to an A, C, G. This T would go to an A. This A would go to a U. So that is as simple as it. It's just changing our letters. That we need to find that base. Like you're kind of looking at it as a base pair. Is that original DNA strand is going to transcribe into mRNA. It's the pairing letters that we've learned from the beginning, except there are no T's, that T's turn into U. Okay, let's jump to going back to our transcription page. So remember, our transcription is taking that DNA and turning it into mRNA. Happens inside the nucleus. It goes from a double-stranded to a single-stranded mRNA. Really, it's the same base pairs, except instead of T's, we use U's. Now, going to the next one is that once we have it transcribed into mRNA, we are going to go through translation, which is going to turn it into a protein, which is made up of our amino acids. So really what we look at here is that our mRNA is split into codons. And this is why, you guys, in the last example, I split it into 3s, because that's what our base pairs are split into when we start looking at, hey, what 3 letters does this amino acid make? Okay, and through the use of tRNA. So codons are linked to amino acids by tRNA, transfer RNA anticodons. Now, tRNA, you guys, doesn't really stick. Our tRNA is just what is going to drop off the amino acid, and it's going to keep going. Okay, so when we look at this, I'm not going to necessarily ask you much about tRNA, but just understanding that it's used in the drop-off. If you watch those Amoeba Sister videos, they are going to give you a good description and animation of how that process works. But what we need to focus on is that once we get that mRNA strand, that single strand with all those letters, well, let's turn it into a protein. And what protein or amino acid is it going to turn into? So when we look here, ribosomes are typically free floating within the cytoplasm. These ribosomes produce proteins. Ribosomes consist of 2 subunits, okay? Each codon to be translated arrives at the A site, okay, with the help of tRNA. So this goes into quite a bit of detail that I'm not going to necessarily focus on because we're just going to go over the basics of the process. Now, this table you will never have to memorize. What this table does is this is where we're going to take our mRNA codons that we have just learned to take from DNA and turn it into mRNA, and we are going to look at what amino acids are going to be produced. For example, if we look here, if I have a UUU or UUC mRNA, and this is very important, that this chart can only be used for mRNA, is that it's going to code for that phenolphthalein. UUA, UUG is going to code for that leucine, or LEU, and we tend to abbreviate them, shorten them so it's not as long. So this is probably the best summary of what we've done. We've taken DNA. We go and take that original DNA strand and go through transcription, which happens inside the nucleus. And remember, we take that original strand. That original strand is going to transcribe into mRNA. Okay? That mRNA, we are going to split into groups of 3. And this is what's new, is that process of translation is going to then occur in the cytoplasm, not the nucleus, and we are going to use the code here, this little chart, to figure out what amino acids there are. So let's look at this first one, UUC. We go back here, okay, we already talked about this one. This mRNA, UUC, is this PHE, and that's the amino acid. Let's go to the next. I'll use a different color. AGA. Okay, remember, it's the mRNA that's been transcribed. We're going to go through translation and turn it into amino acid. This AGA is right here. And the ARG is our amino acid. So on and so on. So this is the best summary, and it goes through a lot of detail, but what we need to be able to do is take the DNA, turn it into mRNA, and figure out what amino acids there are. Okay, let's jump to our practice. We've taken the DNA strand and turned it into mRNA already. So remember, you always work from the original DNA, turn it into mRNA, and now let's go through translation and turn it into amino acid. So how do we do this? We're going to take this codon, GGA, and we are going to go to our chart. GGA is right here, which is our GLY amino acid. And we record that under that codon. So let's go on to AUG and look at our amino acid chart. Okay, our AUG is right here, and it's a MET amino acid. And we record that MET underneath. Our next one is our AAU. And we are going to go here to find our AAU, which is our ASN amino acid. Okay, so let's go to our next one, AUU. And we go here and we find our AUU, which is our ILE amino acid. And we record that right here. Okay, our next one is our ACG. Our ACG amino acid is right here, and that is our THR amino. And I record that THR. So I will let you guys on your own finish these two, because that one's not a complete one. But the important thing here, guys, is that we have taken a DNA, we have turned it into our mRNA, and we have then turned it into an amino acid. So just to kind of give us a bit of an idea is that our DNA goes to here. In order to get mRNA, I have to start with DNA. mRNA can then turn into the amino acids. So it's kind of like a flow chart. So now we can also work backwards. Okay, if I were to give you an amino acid and we start there, and I ask you, well, what would the corresponding DNA strand be? Well, we can also start with our amino acids, work towards our mRNA, and then in towards our DNA. So, but we have to understand that we cannot just take an amino acid and directly figure out what a DNA is. That this mRNA is important because it acts as that stepping stone. Now, let's look at practice 2. How many nucleotides are required to code for the following sequence of amino acids? So, when we look at this, what we're looking at is how many nucleotides. Is that if we look at something like LEU, and we're going to find that on our amino acid chart. Okay, so if we have our LEU, this is all of these. Right in, oops, here's that. Okay, our LEUs are all right here. So what this is getting at is, in theory, to get that LEU amino acid, I can have any of these mRNA codons. I can have any of this. I can have UUA, UUG, CUU, etc., etc. So when we look at that question, is that we can have any of these combinations as the mRNA